everybody, and welcome to the first webinar in the Microbiome online series. My name is Diana, and it is my pleasure to be introducing you to our speakers today. But before I do, I have a couple of very quick announcements to make. First, I want to say a huge thank you to the speakers for making themselves available to present today, and to our sponsors for the series for making this possible. This webinar is being sponsored by Metabolon. Please feel free to use the chat and questions tab to interact with our speakers. This is an interactive webinar, and I will hand it over Ruth Ann to start introducing yourself.
Hi, I'm Ruth Ann Luna. It's morning for me here, so I'm in Houston, Texas. I am Director of Medical Metagenomics at the Texas Children's Microbiome Center at Texas Children's Hospital, and also Assistant Professor at Baylor College of Medicine. We do a variety of next-generation sequencing-based applications, both clinically and translational research, so you'll hear more about that in a little bit.
Thank you. Brian?
Yes, thank you. Hi, everybody. My name is Brian Kepler. I'm the Director of Discovery and Translational Sciences at Metabolon. We're located and headquartered in Research Triangle Park, North Carolina, but we have a global presence, and we provide global untargeted biochemical profiling, full comprehensive look at metabolomics, biochemicals, metabolites, as well as targeted assays. It's my pleasure to join the panel today and talk to you about the applications and the importance of folding in metabolomics in microbiome research.
Thank you. And before I head it over to Elliot, Connie is unable to join us live today, but she's got an amazing talk recorded for you on using multi-omics for studying Crohn's disease. But now I'm going to head it over to Elliot to introduce himself and to kick off the presentations.
Okay, hi everybody. I'm Elliot Friedman. I am at the University of Pennsylvania. I hold positions in the Penn Center for Nutritional Science and Medicine, as well as the Penn CHOP microbiome program, where I run a targeted metabolomics facility that also does microbial culturing. So I'm going to talk to you today about the intersection between microbiome and its host via metabolomics. Since I'm the first speaker, I'll give a brief introduction, but as we're all here to discuss the microbiome, I'll go through this pretty quickly. But the human microbiome is comprised of bacteria, viruses, archaea, and other eukaryotes. And there are distinct microbiomes by body site. Some of the more well-studied ones are the gut, lungs, skin, mucosa, etc. In particular, the gut microbiota is home to approximately 100 trillion bacterial cells. And as we move longitudinally through the GI tract from stomach to rectum, we increase in microbial density, reaching a maximal density of about 10 to the 11th to 10 to the 12 cells per gram of luminal material in the colon. And although there are a large number of species that are present and our culturing techniques are rapidly improving, but many of these organisms are still uncharacterized in defined pure culture systems where they've been able to be cultured in the lab. So for that reason, we generally employ multi-omics technologies to study both the microbiome and its host, as depicted on this slide here, moving from left to right, starting with the sequencing of the DNA genome, looking at the DNA that both the host and the microbiome have. We can look at the epigenome to look at DNA methylation and other DNA-based modifications. And moving on to the transcriptome, we can look at the RNA that are actually being transcribed and the proteins that they make with the proteomics, looking at peptides and proteins. And we can also look at the metabolome, which is looking at the small molecules that are used both as substrates and that are produced by the microbiome and host. And these can include lipids, carbohydrates, and amino acids, among others. And the goal here is to use one or more of these technologies to understand the phenotype and the relationship between the microbiome and its host. So let's focus specifically on metabolomics, and you'll obviously hear more about this from Brian. But just to give an overview, something that I find is generally helpful is to classify these metabolomics into two separate groups. So on one hand, we have untargeted metabolomics. It's also called global or discovery metabolomics. It's on the left here, as opposed to targeted metabolomics, shown on the right. So I'll go through a few differences between these. Generally, untargeted metabolomics approaches use liquid or gas chromatography coupled to mass spectrometry as a detector. And targeted metabolomics can also employ these hardware configurations, but can also use traditional liquid chromatography, gas chromatography, or NMR. And in an untargeted approach, the samples are often run through different hardware configurations, so hydrophilic or hydrophobic columns and positive and negative ion modes for mass spectrometry. In a targeted workflow, samples are run on a single hardware configuration that are designed to look at a specific group of chemical analytes. In an untargeted approach, the mass spectra and sometimes the retention time of the spectral features are matched against either in-house subscription or publicly available databases to take the spectral patterns and sometimes their retention times and use them to either definitively or putatively identify these spectral features, these analytes, as specific chemical compounds. In a targeted workflow, the analytes are identified using analytical standards, so actual pure versions of these chemicals that have been run on the same exact hardware, and usually they are run during the same machine run. In an untargeted approach, the data is reported as raw chromatographic output, so area under the chromatographic or spectral curve, and you get relative abundance. So what this means is that if you look at metabolite A, you can say that metabolite A is higher or lower in a control group versus a disease. And you can do the same thing for metabolite B, but you cannot actually compare metabolite A versus metabolite B, either within the control or the disease groups. In a targeted workflow, the data is reported as absolute quantification, so volumetric or mass-based concentration. And you can make the similar comparison, metabolite A, control versus disease, but you can also compare the levels of metabolite A and metabolite B within and between groups. And finally, just to note the cost and the necessary post-analytical statistics, an untargeted metabolomic approach is usually more expensive and often requires more rigorous statistical approaches, as you, instead of having tens or hundreds of metabolites, you likely have thousands or tens of thousands. In a targeted workflow, this is usually less expensive, although that can depend exactly on your specific analytes. And the analysis can be simpler because you can be looking at anywhere from a single targeted metabolite to 10 or 100 metabolites. So for the rest of the talk, I'll bring you through two studies that we've done at Penn and Children's Hospital. And these employ one employs untargeted metabolomics while the other employs targeted metabolomics, to give you an idea of how we use these to get at the microbiome and the host. So the first study is called the FARM study for food and resulting microbial metabolites. And up here in the upper right, you have a schematic of the study design. So this study involved 30 healthy human subject volunteers, 20 of whom were omnivores and 10 of whom were vegans. The 20 omnivores were randomized to either receive a control omnivore diet or to receive exclusive enteral nutrition, which is a purely liquid diet that's used for the management of inflammatory bowel disease. The vegans, I'll note, were outpatients because based on our previous studies, we've actually found that vegans are have very tractable diets, and we don't actually need to rigorously control them as much as we do for omnivores. There were 4 phases of this study. In the first phase, the dietary phase, essentially the omnivores, we observed the effect of the omnivores and the exclusive enteral changing their diets from their standard outpatient diets, and the vegans stayed the same. For days 6 through 8, these subjects all received an antibiotics and polyethylene glycol purge that served to wipe out or drastically decrease the load of the microbiome in the gut. And you can see that here as a percentage of reads. Dark blue is non-host or bacterial microbiome, and light blue is host. And you can see that there's a rapid and substantial decrease following this antibiotics and PEG flush. And we confirm this via qPCR and culturing. And essentially, this intervention causes a decrease in bacterial load of about 5 logs. We then observe the recovery biome and its metabolome, and we subdivided this into 2 stages, early and late. The microbiome composition and the changes are shown on the bottom portion of this slide on principal coordinate analysis, so separated by the 3 dietary groups. And each dot here is an individual subject and an individual day. And the study days are shaded from green to purple. And so what you can see is that in both the omnivore and vegan groups, they start on the left side. In the upper left, they move over to the right following the intervention. And then they actually, at the very end, start to move back, or the arrows start to move back towards the beginning. It's drastically different for the exclusive enteral nutrition group. First thing we can see is that there's actually an effect of the diet intervention. The green dots, there's a significant move downward. And then there's actually these subjects, their microbiomes end up in a completely different place. And this is essentially due to the lack of fiber in this exclusive enteral nutrition formula. And I won't go into the further microbiome results for the sake of time here, but there are some observations that we were able to make in this study. We did use an untargeted metabolomics approach to look at both the fecal and plasma metabolome in this study over time. And in general, we saw robust changes in the fecal metabolome that is shown here on the same type of principal coordinate analysis. And you can see that for each of these diets, there's significant alterations over the course of the study. And to be able to handle the number of metabolites, which was about 6,000 that were able to be matched to chemical IDs based on databases, we employed an analytical strategy where for each group, we looked at the change in each metabolite in each portion of this study. So from day 0 to day 5 to day 9 to day 12, and day 12 to day 15. And for each of these 4 intervals, we used a statistical approach to define whether the metabolites were increasing, stayed the same, or went down. And we assigned these numerical codes of 1, 3 respectively. And so then what we're able to do is actually look at the distribution of the patterns of these metabolites. We get 4-digit codes. So for example, a code 2222 would mean that that metabolite did not change in throughout this entire study within that group. And so in panels C and D here, what you're looking at is the distribution of metabolites by group with specific codes, and then the overall count of metabolites with specific codes in panel D. And the omnivore group is red, the vegans are green, and the exclusive enteral nutrition group is blue. So one thing to note is actually that there's the largest group of metabolites are 2222. And so this robust intervention of both dietary and antibiotics, polyethylene glycol, actually did not affect a significant number of metabolites here. And then look at other patterns. And one thing I'll note here is that all of the green, so those are the vegan groups, have a code that begins with 2. And if you remember, I said that the vegans actually have quite tractable diets in an everyday outpatient setting. And so we did not change their diets from day 0 to day 5. And thus, we observed no changes in metabolites. So beyond those broad observations, what we can do is focus in on specific groups of metabolites. And based on our microbiome and previous observations, we focused on amino acids and amino acid metabolites that are produced by the microbiome. And so just to orient you here, what we're looking at is untargeted metabolomics data from amino acids in panel A and amino acid metabolites in panel B. And here we've shown the changes in the antibiotics PEG intervention and in the early recovery stages by group. Blue indicates that the metabolites went up, and red indicates they went down. And we observed these in both plasma and stool. Downward leftward facing triangle indicates plasma, up and right facing indicates stool. So generally what we see is that amino acids, which are utilized by the microbiome, when you wipe out the microbiome or drastically decrease the microbiome, we see these increase in the stool and to a lesser extent in the plasma. And these are recovered, that we see they go back down during the recovery of the microbiome in the omnivore group, but not in the exclusive enteral nutrition group. And we observed the opposite pattern here for amino acid metabolites. And a few of these, but only a few, the ones with asterisks here, so 5, have been previously linked to the microbe to be produced by the microbiome. And so what we're able to say from this is that we've actually identified 27 novel metabolites that, based on their patterns following this dietary and antibiotics PEG intervention, appear to be a product of the human gut microbiota metabolism. So this study is an example of using untargeted metabolomics to probe the relationship between the microbiome and its host. The key takeaways are that we identified dietary effects on firmicutes that alter carbohydrate and amino acid gut metabolome, and the effect of diet-based microbiota metabolites on the plasma metabolome is fairly modest, and that data I didn't go into detail for the sake of time, but you can feel free to look at the paper and contact me about it if you'd like. And finally, we identified 27 novel metabolites that appear to be a product of the human gut microbiota metabolism. So the second study that I'll give you as an example here is is a study in which we looked at the bidirectional relationship between bacteria and bile acids in the gut. And here we used a targeted metabolomics approach. So bile acids are compounds that are produced by the host in the liver, and they're excreted into the small intestine following food consumption. They're then subject to modification that occurs exclusively by the gut microbiota. So the first step is a deconjugation, or removal of glycine and taurine conjugates, host-produced bile acids, by an enzyme called bile salt hydrolase. And this enzyme is present in a number of gut microbes that are across different phyla. The unconjugated bile acids are then transformed by via 7-alpha dehydroxylation by a group of genes in the bioperon, and these genes are exclusive to a number of taxa in the Clostridium species, or as Clostridium genus. So interestingly, bile acids also have an effect on bacteria. So gram-positive bacteria are actually more sensitive to bile acids than gram-negative bacteria, but they have toxic effects. And we use this in a microbiology setting to actually selectively culture gram-positive versus gram-negative organisms. This bile acid toxicity to bacteria is multifactorial and involves membrane effects and DNA damage, among others. And these bile salt hydrolases that I mentioned are really only found in organisms that inhabit the intestinal tract of mammals, and it's believed they serve to enhance the colonization efficiency of these organisms. So we looked at the interplay between the microbiome and bile acids in a controlled human subject study examining the effect of a synthetic bile acid analog called obeticholic acid, which inhibits bile acid synthesis through activation of the nuclear hormone receptor FXR, farnesoid X receptor, in the distal small intestine. And so we looked at the physiological effect of this drug by looking at this chemical called C4 that's an intermediate in the biochemical synthesis of bile acids from cholesterol. And here we observe a rapid or a robust decrease in this biochemical intermediate on day 16 of this study, which is when the subjects were on drug for a maximal amount of time. So there's a baseline point at day 1, a on drug point at day 16, and then they're off drug again at day 37. And when we look at the fecal microbiome, we actually see an inverse pattern here shown here in the most significant organism, Streptococcus thermophilus. But there are actually 4 other organisms listed here that had FDR significant associations with OCA use, and I'll note that they're all gram-positive organisms. And so our hypothesis was that these are all organisms generally associated with food and or the oral and small intestinal microbiome, and they're actually not normally seen in feces. So to understand this, we took these organisms, we cultured them in the laboratory, and we looked at the inhibitory effect of endogenous bile acids, glycokenogenic acid and glycocholic acid, as well as the drug obeticholic acid. And so just to orient you here, we're looking at percent reduction in bacterial growth, where a darker redder color indicates a more inhibition. And in general, what you see is that these endogenous bile acids have significant inhibitory effects on most of these organisms, while the drug obeticholic acid does not. And these were both run at what we observed to be physiologically relevant levels in the human small intestine. So this again strengthened our hypothesis that the bacterial taxonomic signature in response to OCA was likely due to decreased endogenous bile acids that result in a signature of small intestinal bacteria in the feces. So we tested this by redoing this study in animals. So we treated mice with obeticholic acid for 2 weeks, and then we collected proximal and distal small intestine contents as well as feces to look at the actual bile acid levels using a targeted metabolomics approach. And I'll note that since we use a targeted metabolomics approach, we can actually combine the different bile acids. So we can look at total bile acids versus primary and secondary bile acids. We're able to do this specifically because we're using a targeted metabolomics approach that gives us molar-based concentrations that allow these to be added together. And so we observe a robust decrease here in the red drug group compared to the control in the vehicle in the proximal small intestine, as well as a less pronounced but still reduction in the distal small intestine. You actually don't see this in the feces. And the reason for this is actually that about 95% of bile acids are reabsorbed in the distal small intestine, and they go through enterohepatic recirculation. And only about 5% come out in the feces. And you can actually see here that although these are in nanomoles per gram versus nanomolar luminal contents, the levels here are about 2 orders of magnitude lower. So I'll note that we also observed an outgrowth of gram-positive Clostridiaceae in the proximal and distal small intestine, and that would be consistent with our idea that this decrease in bile acids leads to an outgrowth of gram-positive bacteria. So basically what we can see is that this targeted metabolomics is a powerful tool for investigating relationships between the gut microbiome, its host, and specific metabolites already known to be linked to the gut microbiome or of specific interest for your study. And here we were able to elucidate the links between bile acids and the small intestinal microbiome. And this is incredibly useful approach. Targeted metabolomics can be used for identification of potential therapeutic and biomarker targets. To bring it back to our previous example, this untargeted approach is really useful for identifying new compounds of interest and really probing the relationship between the microbiome and its host. And so I'll simply conclude by saying it's very important to identify and apply the proper technologies, in this case metabolomics technologies, to microbiome studies so that you get the most applicable results for your study. So with that, I'd like to thank our group at University of Pennsylvania Children's Hospital, as well as our collaborators on the second study at Intercept Pharmaceuticals, and our funding, which includes the Crohn's and Colitis Foundation. Thanks very much for your time.
All right, thanks, Elliot. Again, my name is Brian Kepler. I'm just Director of Discovery and Translational Sciences at Metabolon. And sort of piggybacking off of Elliot's talk, again, I just want to use my talk to emphasize the power of metabolomics in microbiome research, right? Metabolomics is really key to attribute the function of the microbiome. You know, what are the metabolites that it is consuming? What are the metabolites that it is excreting? And really the main takeaways I want to convey today are, you know, not just the advantages of metabolomics and microbiome research, but also, you know, why metabolomics in addition to other omics, genomics, proteomics, transcriptomics. You know, metabolomics isn't meant to supplant these others. It's meant to be complementary and marry very well with them. And along with my case studies, another point I'd like to make is just how far metabolomics has come, you know, over the last, you know, let's say 20 years since Metabolon came to be, and even in the last 10 years. You know, the quality of the data, the ability to interpret the data, and also the way that metabolomics can be employed now to support regulatory approvals, as demonstrated in a mechanism of action example that I'll give as part of a company's submission, and I'll give a case study for that. So following on with, again, what Elliot touched on already, why metabolomics, right? Well, metabolomics incorporates all the elements of a biological system, right? It actually provides that definitive representation of the phenotype. It's the omics that's closest to the phenotype, right? And with the central dogma of genomics, transcriptomics, and proteomics, really metabolomics is able to not only take into account those other central dogma elements, but also things like the microbiome and the environment. So the environment being our diet, obviously, as what we consume is digested in the microbiome and has a profound impact on that, and then can have a profound impact distally with it throughout the body based on the metabolites it produces. Do we exercise? Where do we live? Do we smoke? What medications do we take? All these different things can really tell the complete story. And that's why, in this talk, I'll focus on, starting out with global metabolomics to really get the most comprehensive view. And, again, genomics, has its place, but genomics tells you about your genes, right? It can give you the likelihood of getting a type of disease, but whereas genes are the key influencer of biology, they don't provide the full picture, right? The onset of every disease involves a biological change, right? When you get a disease, your genes aren't changing, it's the biology, right? So, at Metabolon, through our different technologies from global untargeted through targeted, we measure the full metabolome from biological samples with the hopes of informing our clients and really communicating what all of this means. So, as I mentioned, to really leave no stone unturned in your R&D efforts, looking through all the omics, to do metabolomics, as I said, there's targeted efforts looking at just short chain fatty acids, just bile acids, right? Those are, of course, two main areas of microbiome emphasized research. But as I hope to convey during this talk, looking, really opening the aperture as far as possible and looking at not just microbially derived metabolites, but metabolites as the host is really the best way to get the fullest picture. So this is a look at all the major superpathways and subpathways of biochemistry in a living organism. And these are all the pathways that Metabolon's global untargeted pathway surveys. So amino acids, carbohydrates and energetics, nucleotides, lipids, as well as those microbiome specific metabolites. And, there was a question I saw in the chat already, how do we distinguish microbiome-derived metabolites from host metabolites? And of course, there's going to be some degree of overlap. Many metabolites are similarly conserved amongst mammals and bacteria and even plants. But bacteria contain enzymes that mammals do not. And, the assimilation of things like aromatic amino acids or choline or diet-derived metabolites such as fiber, right? The bacteria contain these enzymes and produce a metabolite. So when we see these metabolites in feces or even recirculated into the plasma, we know that it's a contribution directly from the microbiome, right? And then these actually can be sent distally throughout the host and be further modified, let's say sulfated, and then actually have detrimental effects. There's case studies that Metabolon's been involved in where it can have toxicity effects or effects leading to CNS disorders or liver dysfunction, things like that. So really, maximizing the value of each experiment, it is important to profile broadly and to look at the host metabolism, the microbiome metabolites, and also those xenobiotics, right? The drugs, the diet-derived metabolites. So I don't want to spend too much time on this, but just a quick overview of how Metabolon does what we do. And this is a similar approach to the untargeted approach that Elliot mentioned. So at Metabolon, to deliver the highest quality results and fully annotated biochemicals, we are involved with our clients from the inception of an idea of a study, right? We like to provide personalized guidance at every step of the collaboration, from study design and planning to powering that success, all the way through interpretation, right? The way we do our untargeted approach, and I'll talk about some targeted case studies at the end, but the untargeted approach, it's a single platform of 4 different methods. They're all ultra high performance liquid chromatography in tandem with mass spec. And the 4 methods have some different columns, for example, or they're optimized for positive ionization or negative ionization. And the reason for 4 methods on one platform, so every sample that we get, whether it's plasma or feces or anything, is split, run across these 4 methods. And the reason is to be as broad as possible, to capture the largest breadth of different classes of biochemicals, because some are going to be polar, some are hydrophilic, hydrophobic, and by doing these 4 methods, we can be as comprehensive as possible. Now, Metabolon's hardware is typical off-the-shelf Thermo Fisher hardware. Now, really the hard part is what comes next, right? This is going to produce hundreds of thousands of ion features per sample, right? So the trick is, when you're looking at metabolomics data, is can you distill that? Can you eliminate all the noise, reduce the data, and switch it from an ion-centric approach, which is just looking at ion features, to a chemocentric approach, which is actually attributing those ion features to a specific metabolite. And that's what we do at Metabolon. It's fully annotated tier 1 identification. So we know the retention time, the mass, the fragmentation. And the way that we do these fully annotated identifications in a global untargeted approach is that we've developed an in-house library, right? A reference library that is truly the world's largest and unmatched in terms of its comprehensiveness. And we are able to identify these and map them to their different pathways, produce a heat map based on the statistics, and then do the interpretation. And the interpretation is really the step beyond the data, right? My team specifically is involved in taking this massive amount of information from spreadsheets and heat maps and trying to narrate a comprehensive and credible story for our clients to further their research discoveries. So the applications of metabolomics is extremely broad, right? I'm going to focus obviously on the microbiome in the slides to come, but we work with clients from the academic realm to government institutions, the NIH, and pharma and biotech and applied markets as well. And whether the objective or application is looking at prediction of disease outcomes or optimizing drug development, patient stratification, biomarker discovery, and mechanism of action elucidation, metabolomics really has a place in all these. And you can see from the slide, there's examples and literature references from all these. But again, to focus on the topic of today and for the audience, really, I want to emphasize not only the importance of folding in metabolomics into your microbiome research, but the ability of Metabolon's technology to help you do so. So a little bit about the host microbiome relationship. Metabolites obviously are the cornerstone of this relationship. I mentioned before, the bugs in your gut or on your skin or wherever it is, they literally consume metabolites and they excrete metabolites. So not doing metabolomics is really a disservice to your microbiome research. Looking at metagenomics, 16S, shotgun sequencing, obviously that's a key step in it as well, but that information is only going to tell you the presence, are the populations changing? Just like proteomics is an important step in normal research, but if you don't know if it's active and if it's actually an active enzyme converting a metabolite substrate to its product, you don't know if it's having a phenotypic effect. So, the metabolomics is really at a focal point of this intersection between the host and the microbiome. And, it's a catalytic wonderland, right? It's driven by vast chemical and enzymatic diversity. There's dietary compounds, industrial and environmental influences, pharmaceutical influences as well. And all these different things, again, can lead to distal effects in the host, influencing disease risk, bioavailability, toxicity, efficacy. So, last thing I wanted to mention is an article in Nature that I referenced in the top corner there, that, it's been found through a number of publications that whereas genetics obviously has a degree of shaping the gut microbiota, it's actually the environment, right? The environment which the metabolome and metabolomics can yield insight into, which dominates over genetics and shaping that gut microbiota. So some limitations of common microbiome research approaches, and really where Metabolon can fill those gaps is shown here. I mentioned, microbial identification, shotgun DNA sequencing, using live biotherapeutics, microbially directed drug development. All these things are good to do and fold into your research and development, but they also have their challenges and limitations. Metabolomics can really identify those metabolites, not only in the feces or in the blood, but really any biological matrix that you can harvest from any living thing, and really address the weaknesses of these current approaches. It's like, putting a puzzle together. If you have a 1,000-piece puzzle, if you're missing some pieces, you're not going to get the full picture. So really, not leaving out the omics that is closest to the phenotype is really key to elucidate and get as granular as can in the picture of your disease or your study design. So before I get into some specific case studies on data, I wanted to share a couple of real seminal papers, some key publications that show the power of metabolomics in furthering microbiome research. This first one is looking at the interplay between the human gut microbiome and host metabolism. This was published in Nature Communications in 2019. And in this study, the metabolomic pathways were found to be associated with 34% of blood and 95% of fecal metabolites, with over 18,000 significant associations, while the different species were only able to show about 3,000 associations. So this study was really able to underline the importance of studying the microbial and metabolic potential rather than focusing purely on taxonomy to find therapeutic and diagnostic targets. The other example here on the right was looking at the blood metabolome and how it can predict gut microbiome alpha diversity in humans. So this is a paper looking at Shannon diversity. And what was found is that 40 diverse plasma metabolites using global untargeted biochemical profiling through Metabolon's technology, those 40 metabolites were able to predict the Shannon diversity, where a wide range of clinical tests and a set of 263 proteins could not. Again, emphasizing the fact that the host metabolome is an important interface between the gut ecosystem and human health. A couple other key papers shown here, again, emphasizes the applicability of metabolomics and the fact that other omics alone may not tell the full story. The one on the left here, published in Nature, was looking at the structure, function, and diversity of a healthy human microbiome. So on the top here, you see the different phyla, the different species. It's hard to see, but you have all the different bacterial species and the different colors. The bottom row here is the metabolic pathways with all the different classes of metabolites. And this was done by the Human Microbiome Project Consortium. And it was found that 242 healthy individuals profiled from that Human Microbiome Project actually revealed diverse phyla, right? You can see from that top row that there's quite a bit of diversity. However, the metabolomic function is pretty similar. In the same regard, we have seen in studies where the phyla are similar. The gut microbiome, let's say the metagenomic, the 16S sequencing is similar between individuals or between disease states or before and after drug, but the metabolomics is different, right? So again, emphasizing that, stopping at metagenomics or stopping at, genomics or proteomics or transcriptomics, again, doesn't tell the complete story, right? Sometimes you might see changes in phyla, no changes in metabolomics. Sometimes you might see no changes in phyla, but changes in metabolomics. And the last one on the right here, this was published in Nature Genetics a few years ago, was regarding the fecal metabolome as a functional readout of the gut microbiome. So again, on the y-axis, we have the bacterial species, and on the x-axis, you have the different classes of metabolites. And by combining these fecal metabolomics with microbial sequencing, in this case 16S profiling, it was able to identify associations with microbial OTUs and taxa. So you can see that just by looking at the metabolites, you can identify whether or not the diversity is penetrant. For example, on the example on the left, you can see associations between the metabolome and the microbiome. You can get estimations into alpha diversity by the Shannon index on the previous slide. All right, now getting into some case studies. I'm going to focus on a few, due to the sake of time, that are targeted assays. So switching gears a little bit. So this was done with a biotech company, and basically using Metabolon's targeted assays, both short-chain fatty acids and bile acids, to gain insights into the mode of action using these pre-developed panels. And in this case, we're dealing with ulcerative colitis, right? An inflammatory bowel disease often characterized by low levels of the beneficial bacteria and high levels of the pathogenic bacteria. And this particular company focused on the formulation of firmicutes and other bacterial species to change the microbiome composition in disease, where that pathogenic bacteria are believed to be causing or contributing to the disease, right? So the compound used, which in this case is SER-287, it's a next generation investigational biologically derived microbiome therapeutic. What they found in this study was that in results from a phase 1b trial, changes in short-chain fatty acids and bile acids were able to confirm the changes that were seen in the microbiome composition. So you see engraftment of SER-287 species in the bar graph on the left. You see metabolite levels in the fecal samples on the right. Now, taking what was learned from this study, there was a phase 2 trial done by the same company using a GCP bile acids assay. SER-109, another drug, a next generation phase 2 microbiome drug to reduce the recurrence of C. diff infection, was used. And the recurrence of C. diff infection occurs in quite a large proportion of patients prior to infection. So looking at this, the engraftment of SER-109 species on the top bar graphs, and then the look at the bile acid correlation with the number of SER-109 species on the bottom. You know, the company, based on this metabolomics data that Metabolon was able to provide, they were able to leverage this global metabolomics to discover these biomarkers and targeted metabolomics of these bile acids to validate their data and satisfy compliance with regulatory standards. You know, the reproducibility, the high quality of the data increased the confidence of how their therapeutic was changing the metabolism, in particular with these bile acids in the GI, which obviously relates to the pathogenesis of C. difficile. So basically allowed the client to have a much clearer picture of the biological activity of the microbial community, really able and prompting this team within this company to reanalyze their phase 1 data and ultimately redesign their phase 3 study in terms of looking at the mechanism of action. And to sum up, again, due to the sake of time, I wasn't able to go through all the different applications and experience that Metabolon has in microbiome research. But on the right here, you see some headlines of a real multitude of successful case studies we've had and publications that span a variety of applications in therapeutic areas. We've looked at asthma, liver cancer, immune checkpoint therapy, things like autism, and how microbiome metabolites can be recirculated into the plasma and then go into the brain and affect an autism spectrum disorder effect. Looking at diet, the ketogenic diet, it can actually, the gut microbiota has an influence on the anti-seizure effect of the ketogenic diet. Type 2 diabetes and glucose intolerance, ALS, NASH, really the list goes on. And I'd be happy to share offline with anybody who would like to contact Metabolon some of these additional case studies and how metabolomics can inform on these. With that, I'd just like to give my appreciation to the other panelists and to the organizers for the opportunity to share today. Thank you.
All right, so moving on, I guess I'm up. So I'm going to take you on a whirlwind tour of how we have utilized multi-omics, really digging into the complex clinical phenotypes, in this case of autism spectrum disorder in children. So we're really focused on the microbiome gut-brain axis in pediatric autism. So to illustrate that, let's take, for example, a child with autism who has difficulty communicating but is experiencing abdominal pain. So rather than being able to verbally communicate that they are in pain, they may in fact resort to behaviors, maybe self-injurious or aggressive behaviors, because behaviors we know can be a very effective form of communication. But as you can imagine, those types of behaviors then increase stress. Stress further exacerbates the GI symptoms, and you see there the start of a vicious cycle. So here we have a 2-week time series case study of a single child with autism where daily stool samples were collected, as well as on the far right, a single stool sample collected from an unaffected sibling. And what you can see here is that in the middle of this 2-week period, you see a shift in the microbiome with a very similar shift happening at the end of that 2-week period. And when we layer on our GI data, you can see that those changes in the microbiome actually coincide with 2 distinct GI episodes, both of which include the report of abdominal pain and one of which also includes the manifestation of significant diarrhea. But if we take this a step further and layer on behavioral data, now we see spikes in self-injurious behavior coinciding with those 2 GI episodes. So this is a great illustration of the microbiome gut-brain axis in action. So I love this slide from a review that was published in 2017 that attempted to visually summarize everything we knew about the gut microbiome in autism to that point. It's important to mention that all of the studies so far have been in children. We're only seeing maybe a couple of studies now that have been in adults with autism, but most of what we're finding would also be relevant to that adult population. But what we knew at this point in time is that there were some studies that failed to find a difference between their ASD group and their control group. However, there were significant issues with the selection of that control group. Many studies only used unaffected siblings as the singular control group, which of course would lead to significant confounders in both shared genetics and shared environment. Other studies actually excluded participants on the basis of GI symptoms, which we would argue is really the population that we're chasing in a lot of this research. The other issue is that very few studies took diet into consideration at all. And we know that diet is a significant driver of the functional microbiome. And then the final point there is that even in the studies that did find differences, everyone seemed to find different differences. And we think we have an explanation for that. And it's what I like to call the invisible problem, which is by and large our greatest limitation in the field of autism research. And that is that there is no one autism, even though we're forced to treat it as such. A diagnosis of autism spectrum disorder is not an effective stratification variable in clinical research because we know that there's extreme heterogeneity in that population. When we migrated from the DSM-4 to DSM-5 criteria, we saw much milder conditions, specifically Asperger's and pervasive developmental disorder not otherwise specified, lumped into that single diagnostic code of autism spectrum disorder. Add to that the fact that we still fail to agree upon a gold standard tool for diagnosis. Some like the ADOS, others like the ADIR, still others will use the CARS. And that diagnosis is still made solely on the basis of behavioral characteristics with no consideration for what we now all recognize as common medical comorbidities. One other point I'd like to mention for those of us that like to dive into the published research is that as it stands now, the severe end of the autism spectrum, these individuals with profound autism, are still significantly underrepresented in the published research. And that's really due to inherent issues in the study design. If you're doing a study that includes a functional MRI component, a child profoundly affected by autism is not going to be able to remain still in the MRI machine, much less follow commands being given to them while in the machine. Even something as quote unquote simple as a blood draw could lead to non-participation by individuals who have profound autism because their caregiver knows that blood draws are highly traumatic for their loved one with autism. So we've been very mindful about how we design our studies to be as inclusive of the entire spectrum as possible. So when we look at how we have created this multi-omic profile, you can see here the various components, and we really wanted to do a deep dive on the clinical phenotype. So looking at the GI profile, we had all of the participants and their families complete a 2-week diary that included instances, frequency, and intensity of pain, as well as information on the stooling pattern. So what is the stool frequency and the stool consistency based on the Bristol stool rating scale. We also had every participant assessed using Rome 3 criteria for the diagnosis of any potential functional GI disorders, and we rated their symptom reports using the GI severity index. Now, we talked about diet and the importance of including diet in these analyses. Yes, we asked the families, is your child on a specialized diet? Specialized diets are actually commonly used in the autism population, but we also wanted a 2-week diet diary to look at not just what the child was offered, but what they actually ate, because many of these children can actually self-restrict themselves into a different dietary category. And then we had a handful of surveys that we utilized to get insight into the behavioral phenotype. Now, I mentioned wanted to be inclusive of the entire spectrum, so in terms of specimens, we required no blood. We only required a single stool specimen that was collected at home and allowed us to characterize both the microbiome and the metabolome. So here is a snapshot of the different behavioral tools that we had these families complete. And if you look on the right, you'll see several subscales that are in bold. And these are the behaviors that we hypothesize would be most closely associated with the presence of abdominal pain in these children. Now, when it came to how we wanted to tackle the dietary information, we wanted to do something meaningful for the families that were going to eventually read this research. And mind you, we've done a lot of things with these diet diaries, but this has really been the most global and potentially the most impactful. A hot topic right now in gut research and microbiome research is looking at food additives and food preservatives. And we've got mounting evidence showing that these new additions to our diets over the past decades are actually associated with increased inflammation and even potentially have a carcinogenic effect. So we've utilized something called the NOVA classification scale. And what you can see here at its extremes are these 2 rows of pictures. The top row being more your whole foods diet. So you've got fresh fruit, whole grains, home cooked meals, and water, whereas the bottom row is sugar and junk food. So there are actually 4 different groups as part of this NOVA classification scale, ranging from 1, which is, again, the whole foods diet, your unprocessed foods, all the way to your ultra-processed foods, which is really, unfortunately, the junk food that most American children sit down to at lunch every day. Now, this is the snapshot of the larger cohort. Again, our 3 groups that we included were children with autism, their unaffected siblings, and unrelated typically developing children. Embedded within each of these 3 groups are also children with GI symptoms and those that potentially met criteria for a functional GI disorder. As expected, we saw the ASD cohort skew toward males. You can see there the ratio 85-15, pretty much what we see in terms of diagnostic criteria in children in this age range as of now. But even knowing that there are GI issues across each of these 3 groups, if you look at the GI-SI, which is the GI severity index, and the FGID there at the red arrow, which is the presence of a functional GI disorder, you see there that the ASD cohort still skews toward the more extreme in terms of severity of GI symptoms and presence of functional GI disorders. Now, initially, we wanted to compare those 3 main groups. And what we saw here, and if you look at this Venn diagram in the bottom right, it's a nice visual snapshot that quantitates the number of shared and unique taxa. Now, what was not all that surprising to us, actually, is that these siblings had their own unique microbial profile. We had seen this previously in looking at children with food allergies and their non-food allergic siblings. So for the basis of this preliminary analysis, we actually set the siblings to the side and then further stratified on the presence or absence of GI symptoms. And as you can see here, there remain a variety of shared and unique taxa across these 4 groups. So what did we see? When we looked at typically developing children compared to children with autism, we saw a variety of correlations with the microbiome differences, factors associated with behavior, their dietary patterns, and GI symptoms. So since this is a very short talk, I'm just going to give you a few examples of some of the connections that we saw with these clinical phenotype characteristics and the microbiome. So looking specifically at behavior, one of the more concerning behavioral issues in children with autism is self-injurious behavior. And it has been hypothesized many times that the presence of self-injurious behavior is closely associated with the presence of abdominal pain, many times undiagnosed abdominal pain. So what we can see here with regard to the microbiome is as self-injurious behavior manifests or increases, we see an association with an increased abundance of Butyricoccus and Vibrio, as well as a decreased abundance of Roseburia. Another interesting factor here is the presence and increase in repetitive behavior, where we saw a significant increase in the amount of Escherichia shigella. Now, of course, most microbiome studies are done by targeting a variable region of the 16S rRNA gene. So the entire 16S rRNA gene does not allow us to differentiate between Escherichia or Shigella. However, we do have whole genome sequencing on this entire cohort that we're currently digging into. So we'll be able to determine if there is in fact a pathogen associated with that significant increase in repetitive behaviors. When we looked at social responsiveness, we did see an inverse relationship where we saw decreases in paraprevotella and increases in akkermansia. Now, looking at diet, this blue bar here that you see in the center are individuals that are on a highly ultra-processed food diet. So this is the junk food again. And so we see increases in both bifidobacterium and actinomyces in those children. Now, really one of our most fundamental questions in this study was, if you took children with autism and typically developing children who had the exact same GI symptoms, could we still differentiate between the two? And the answer here is yes. As you see here, a couple of examples of significant decreased abundance of both Coprobacillus and Faecalibacterium in our ASD plus GI group compared to our TD plus GI group. Now, looking solely at our ASD cohort, and of course, again, we've hypothesized that GI symptoms would be closely associated with behavior, and we saw across our multiple survey tools that was absolutely correct. So across the board, we saw increased behaviors as measured by these tools associated with that ASD plus GI group. But you can see here a handful of those that met statistical significance as well. When you look down this list, it's not surprising to see some of these behavioral characteristics associated with abdominal pain. With the CSP is the child sensory profile, so sensory issues associated with avoiding behaviors, with auditory sensitivity, with body positioning, obviously something we would absolutely think would be associated with abdominal pain. And then looking at the bottom at the RBS, which is the repetitive behavior survey, again, behaviors like self-injurious and compulsive behaviors that we would expect to be associated with abdominal pain, as well as the need for rituals and sameness. Now, I have one other piece that I want to share with you today, and this is a bit of an accidental controlled environment pilot study. Now, I mentioned before that we really were striving to be as inclusive of the entire spectrum as possible. So to make sure we included those who had profound autism, we actually worked with a residential facility. Of course, these children being more severe by nature of them needing to be inpatient. What we didn't realize is that part of the residential treatment program includes an in-depth dietary intervention. So when we first began the analysis of the large cohort, these children were outliers. So all of the data that I've shown you thus far does not include this residential population. But what we did is that we dipped back into that larger pot of that larger cohort and age and sex matched a subset of children with autism and typically developing children to match this residential cohort. Now, this residential cohort having autism, obviously, and being more severely impacted when they were admitted into this facility, were then showing significant improvements in both GI symptoms and behavior compared to the ASD participants in the larger cohort. And these changes in that ASD phenotype were associated with parallel changes in both the microbiome and the metabolome. Now, again, the snapshot of this residential cohort, it did skew towards a little bit of an older adolescent age, again, by nature of them needing to be inpatient. But if you look at this first red arrow, you can actually see all of the different criteria that are related to GI symptom severity. And you see that this residential cohort is actually doing much better with regard to their GI symptoms. The other thing to point out here is that second red arrow where you see the NOVA majority. So this is related to their dietary patterns. So So with the residential facility, we see most of them on an unprocessed food diet, some of them meeting the criteria for a processed food diet, and a few of them meeting criteria for an ultra-processed food diet. Now, the question would be, if they're on this in-depth dietary intervention, how did you capture kids that had the net criteria from an ultra-processed food diet? So even though our food diary covers 2 weeks, for the purpose of the NOVA classification, we actually took the 3 days prior to collection of the stool specimen. And some of these children actually go home for the weekend to spend time with their families, at which point their diet diary indicated that they were on an ultra-processed food diet for those days previous to the stool collection. So that's how they got classified there. But again, that cohort, in stark contrast to the next column where you can see the non-residential ASD, the vast majority of these children were on a highly ultra-processed food diet. So as you can see here, a snapshot of what we saw with the comparison of that residential facility, CFD. As you can see, we saw high-level differences, phylum-level differences, where we saw an increase in Bacteroidetes and a decrease in actinobacteria in that residential cohort compared to both ASD in the larger cohort and typically developing matched kids. We did do global metabolomics, which I won't have time to go into great detail about here, and we did it through Metabolon, and we were able to merge data from a variety of runs over a 3-year period. So we were very happy with how all of that turned out. We had more data than we knew what to do with, and we're still sort of sifting through all of it. But there were some very interesting things we saw when we compared this residential cohort with both the metabolomic and lipidomic data. Most of the difference we saw, we could associate with these differences in dietary patterns. So really confirm that difference in diet. With regard to metabolites, we saw increases in brilliant blue, in sucralose, in erythritol in the non-residential ASD population compared to the ASD population. We also saw increases in both histidine and tryptophan in that ASD cohort compared to the residential population that was on that unprocessed diet. Lots more to dig into there, though. Again, as I mentioned, we do have a lot left to do. We're also going to be able to compare to specialized cohorts. So we have a cohort of girls with Rett syndrome as well as their unaffected sisters. And that has actually been submitted for publication already. And we also have a form of syndromic autism, so specifically Phelan-McDermid syndrome, that we'll be able to compare to this larger cohort, which is mostly idiopathic autism. As I mentioned before, we have extensive dietary data. So we're now quantitating dietary variety to really tackle the picky eating conundrum that's a significant issue in autism. We've characterized the fungal community, and as I mentioned before, we have full shotgun sequencing data that's allowing us to dig into a complete metagenomic analysis, again allowing us to potentially identify important bacterial species and even potential strains. When we collected the stool specimen, we also collected a buccal swab. So we've just recently completed sequencing on the oral microbiome in this large cohort as well, and haven't even had a chance to dig into the data yet. All of these data we expect to help us refine the characteristics of what we see as emerging subgroups in this larger spectrum of autism, and to help us validate these potential subtypes. These subtypes that are based on these complex clinical phenotypes are going to be hugely useful for more effective targeted recruitment for future microbially mediated intervention trials. We do have ongoing studies looking at the effect of both probiotics and antibiotics on GI symptoms and the core behavioral symptoms of autism. So to quickly summarize, if we look at this large comprehensive study, there is no single microbe or group of microbes that is associated solely with a diagnosis of autism spectrum disorder. It's only when we dig in to these complex clinical phenotypes that we can effectively stratify these cohorts of children with autism. We can see now that the presence of GI symptoms is strongly associated with specific behavioral patterns, but we also need to address the fact that diet, age, these behavioral and GI profiles, and even medications all play highly crucial roles in the shaping of the functional microbiome. We do see these microbes and metabolites that are correlating strongly with behavioral variables whose intensity is associated with the presence of GI symptoms. And that on the same ultra-processed food diet, ASD plus GI children still have increased GI and behavioral scores compared to the TD plus GI group. And then finally, we have this emerging evidence that perhaps this unprocessed diet in the residential cohort is associated or even causative for the decrease in GI and behavioral symptoms, as well as the parallel changes in the microbiome and metabolome. So with that, I'll thank my colleagues at the Texas Children's Microbiome Center, and most importantly, my Medical Metagenomics team, as well as our collaborators who helped to recruit for this larger cohort at Nationwide Children's, UT Southwestern, and Columbia University.
Thank you very much, Ruth. And with that, I just realized the time. This hour has gone so quickly, but we still have one final presentation left. So I'm now going to give it to Connie Ha, who will be talking about lots of multi-omic analysis in Crohn's disease.
All right. Thank you for the introduction. So I'm Connie, and I am a postdoctoral scientist at Cedars-Sinai Medical Center. And we are a microbiome lab studying how microbes affect inflammatory bowel disease progression. And so today I'm going to talk about how we use a combination of next generation sequencing tools to investigate a medical mystery specifically occurs in Crohn's disease. And so for researchers who are interested in exploring host microbe interactions in the disease of interest, one of the biggest challenge is how to identify the key players within a complex microbial community that has trillions of microbial cells within the gut, that spans over thousands of species. How do we really identify the key culprit that affects the disease progression? And at the same time, we also need to consider which host cells are actually responding to these microbial members of the gut and actually precipitate the progressions of disease. And so this is the strategy that we use in the lab. We use several high-throughput sequencing techniques to identify microbial as well as the host signatures in disease states. And these approaches actually help us narrow down the potential microbial as well as host players. And then we can actually start to tease out the potential relationship in vitro or in vivo. And that might lead to mechanistic insights. And so for the rest of the presentation, I'm going to demonstrate how this strategy helps us to better understand the mechanistic drivers of extra-intestinal manifestation of inflammatory bowel diseases. And so inflammatory bowel diseases, there are two main subtypes, Crohn's diseases and ulcerative colitis. And they differ in terms of where inflammation occurs. Crohn's disease have patchy inflammations throughout the intestinal tract, whereas ulcerative colitis, they tend to localize in the large bowel. But what's really interesting about the Crohn's disease is that for some patients, they actually have additional disorders or symptoms outside of their gut, and we call these extra-intestinal manifestation. And one of these extra-intestinal manifestation is called creeping fat. And you only really see that in Crohn's disease, and you don't see that in ulcerative colitis at all. So what exactly is creeping fat? And so we've got an image of creeping fat here on the top left panel. So creeping fat is essentially this abnormal fat development that just wraps around the inflamed gut. And in here, and in the image below, you'll see adjacent intestinal tissue from the same patient. And so this gut tissue doesn't have inflammation, and it doesn't have the unusual fat wrapping. And so if you cut these intestinal sections across, you would find that in the inflamed area where creeping fat occurs, this mesenteric adipose tissue actually wraps around the entire circumference of the gut. And so we don't really know whether this fat wrapping is actually is making the disease or inflammation worse, or is actually a protective mechanism for the host. And so at the molecular level, this fat wrapping is actually very different from obesity. And in obesity, you usually define fat expansion as hypertrophy, adipose expansion, where the fat cells get bigger. But then in creeping fat, you actually have a lot of these fat cells, and these fat cells don't essentially expand the way that you would see in obesity. And so what we are interested in is what's actually driving this development of creeping fat in Crohn's disease. Now, being in the microbiome lab, and that this fat tissue is so close to the gut, our hypothesis is that perhaps this creeping fat is really driven by microbes, specifically microbes that originate from the gut. And during inflammation, it actually migrates to the adipose tissue. And we think that maybe this migration of microbial cells to the fat is really triggering a lot of host responses that lead to this creeping fat development. And so the entire story, you can actually look at it in our recently published paper. But in today's talk, what I'm going to highlight is how we use various types of sequencing techniques to really get to the mechanistic insights. And with creeping fat, there actually isn't a robust animal model that really recapitulates all the aspects of creeping fat. So we actually have to turn to surgical specimens to really get to the bottom of what kind of microbial signals might be there and what kind of host cells are involved in the creeping fat development. So we obtained creeping fat tissue as well as healthy adipose tissue and the underlying intestinal tissues from Crohn's disease patients. And we also obtained additional adipose as well as intestinal tissues from ulcerative colitis, which represents a chronic inflammation state, but without creeping fat. So specimens from Crohn's disease and ulcerative colitis can actually help us tease apart what microbial signals are truly reflective of creeping fat and what's actually reflective of chronic inflammation in general. And then we also have healthy tissues that that we use as no inflammation controls. So we first used metagenomics to really explore if there are bacterial signals that are present in adipose tissue, and if there are, what kind of functions the microbial community might possess within the adipose tissue. So we performed shotgun metagenomics on adipose tissue and the underlying mucosal specimens from Crohn's disease and our healthy tissues. And we found that a lot of the bacterial reads actually align with the reads that we saw in stool samples. And so this suggests to us that the mesenteric adipose tissue, they have bacterial reads, and that these bacterial reads, they closely resemble the stool community. So it suggests to us that the community in mesenteric fat is of the intestinal origin. And then we wanted to figure out what sort of function that these communities might possess in the fat. And so we looked at the metabolic pathway, and we found that the Crohn's disease communities have a very different set of genes compared to the healthy specimens. In particular, the healthy specimens are very good at performing fiber fermentation that can lead to, short-chain fatty acids. But with the Crohn's disease community, especially those that were found in the creeping fat, they are geared towards sulfur metabolism and so forth. And so that with the metagenomics data set, what we found is that, yes, there is microbial translocation, and it also occurs in healthy tissue cells. It's not just in Crohn's, but then the microbial function is actually very different in Crohn's versus healthy. And so after that, we decided to expand our cohort and really look more deeply into the fungal and also bacterial groups that might be contributing to this creeping fat phenotype. So we obtained adipose tissues and also the mucosal scrapings from Crohn's disease patients and ulcerative colitis, and performed fungal ITS sequencing as well as the bacterial 16S sequencing. And so the creeping fat group is marked in black here. And so you can start to see that there are some microbial patterns that can distinguish creeping fat, and that some microbial patterns are reflective of the disease state, whether it's Crohn's or colitis. Now, the readouts that we got from ITS and 16S sequencing, we got a lot of potential microbial partners that might be involved in creeping fat. And the list is very extensive, but what we really want to get to the bottom of is which microbes are actually functionally involved in disease development. And we feel that with sequencing, it actually captures signals from both live and dead cells. And so some of the things that we captured are probably artifact and maybe not fully relevant in disease progression. So we really want to get to the bottom of the microbial fraction that are metabolically active and functional in the disease state. And to get to that question, we actually performed microbial cultivation. So we obtained fat tissues from our cohort, we homogenized it and played it out in a range of media, and hoping to see microbial growth and see which microbes are actually alive or survived within these tissue contexts. And to our surprise, there were actually a lot of microbial species that were alive and present in various adipose tissue types. And in creeping fat, we actually found several microbes that are exclusive to the creeping fat tissue, but not found in other fat tissues. And to us, this is a really interesting signature of creeping fat. And what's really interesting to us is the organism Clostridium inoculum, because it's present in almost all of the creeping fat cases. And in cases that it didn't occur, we actually are able to isolate the close relatives of Clostridium inoculum. So we think that this bacterial group is linked to creeping fat development in some ways. Now, during this microbial isolation process, I got a lot of C. inoculum isolates. And so they are mainly isolated from creeping fat, but I also found quite a few C. inoculum isolates in the intestinal mucosa. So I was very interested to better understand whether there are actually strain variations within this C. inoculum group. And just by looking at the colonies, you can tell that they are different. And so does that translate to functional differences? And to answer that, what I did was to perform whole genome sequencing of all the C. inoculum isolates that I have curated through this study. And so what I found was that the C. inoculum that were isolated from the gut, they have a different subset of genes compared to the adipose or creeping fat derived C. inoculum isolates. What we found that were particularly of interest is that the C. inoculum that were from creeping fat, they have more genes that are related to immunomodulation. And so we think that the creeping fat actually selects for strains that are very capable of shaping host defenses, and maybe this is the reason why they're able to survive in the adipose compartment. And now that we've characterized the microbial signatures and we found a really good candidate that might be contributing to disease development. But what about the host sites? Now, the mesenteric fat, it is an adipose tissue, and most people tend to think that fat tissue is just an energy storage compartment. But the reality is the adipose tissue actually comprises of multiple cell populations. It's not just fat cells. There are stem cells and also immune cells within this compartment. So we really want to figure out which cells might be reacting to the microbial signals, such as signals from Clostridium inoculum, and how do they interact with each other. And to figure out which which host populations are involved, we used single-cell RNA sequencing, and we used this technique to characterize the stromal-vascular cells within creeping fat and other adipose tissue, to figure out what cell populations are there and to figure out their gene expression profiles. And we found in total 11 cell clusters in creeping fat tissues and as well as the other adipose tissue. And what's really interesting is that when we start to differentiate where the cells came from, whether it's from the creeping fat or from the adjacent healthy fat, we begin to see that those adipose tissue actually have a very distinct cellular composition. So in creeping fat, we have these very distinct progenitor cell types that were not found in normal fat tissues. And we also see a difference in the macrophage population. So there are a lot more macrophages in creeping fat compared to the other fat tissues. And at the transcriptional level, a lot of these cell types are actually upregulated in wound healing response, and there are a lot of tissue remodeling genes that are being upregulated in immune cells as well as these progenitor cells in creeping fat. And so what you can do after figuring out the microbial candidates and the host candidates is that you can actually put C. inoculum into a gnotobiotic mouse model and to figure out whether Clostridium inoculum can actually translocate in vivo and how it affects host physiology. And here in this experiment, we found that C. inoculum can actually increase fat deposition around the gut. And it's just a very localized response. It doesn't actually change the overall weight. It just increase adipose expansion just around the intestinal area. And also what we were able to do through these sequencing techniques is to actually start to pair up the C. inoculum with other cell populations that we've identified from the single cell data. So we co-cultured C. inoculum with macrophages to see if it actually affects the polarization state and see whether it can actually upregulate this wound healing response. And so after a series of experiments, this is the model that we came up with. So we found that C. inoculum can translocate from the intestinal lumen to the mesenteric fat tissue. And in this mesenteric fat tissue compartment, there are macrophages. And we think that C. inoculum specifically polarized this macrophage to an M2 state, which is very common when there is a wound healing response. And so we think that with this M2 macrophage response, it actually leads to signaling to the progenitor cell types, and that leads to extracellular matrix deposition as well as tissue remodeling. And this is essentially what the creeping fat phenotype is. There's a lot of fat expansion, and there's a lot of fibrosis that occurs. And so to conclude this talk, I'd like to acknowledge all the people in the lab, my PI, Suzanne Defcoda, for allowing me to run the study, as well as the collaborators who actually helped us along the way. And I can be reached through Twitter, and also our lab website is here. So, if our work is of interest to anyone in the audience, you guys can reach out to us. And this is the end of my talk.
Thank you very much, Connie. So I'm now going to invite the speakers back on stage to join me. And I want to say a huge thank you also for answering the questions while we were presenting. We have been influxed with questions, and I think this is probably the fastest hour and a half of my week so far. It's completely just gone really quickly. So most of the questions have been answered. I think in terms of what we should kind of just closing remarks, obviously this webinar covered metabolomics as a kind of focus from Elliot and Brian, and then some multi-omics from Ruth Ann as well. What is the future, what is needed to kind of move forward within microbiome sequencing and to kind of get these complex interactions clearer from researchers? Elliot, could you answer? It's a very big question.
Yeah, it's a big question. I mean, it requires bringing all of the technologies to bear. And unfortunately, that means you have to have a lot of resources and a lot of expertise. I think building collaborative teams is really important. So I think I mentioned this in the chat, but in a response to someone's question, we work not only with computational biologists from that perspective, but also with a dedicated biostatistics group that helps us develop and apply specific statistical techniques to understand what we actually want to look at. And it sounds it maybe sounds really like basic or simple, but using the right techniques, the right statistics to actually answer the question you want to answer is really important. And, from a metabolomics perspective, I'll just say that I think this is generally the feeling in the field that untargeted metabolomics is certainly the most powerful way to identify new relationships, but to understand what's going on, you really almost need to follow it up with metabolomics, because if you want to be able to move into a wet bench or animal model or even an interventional study, you actually need to know what those doses are. And the untargeted metabolomics is just not going to tell you that. On the other hand, it's impossible to identify new targets without taking a discovery approach. So it's kind of this catch-22 that there's no way to get around. You have to employ both of these technologies.
Ruth Ann, do you have anything to add to that?
No, just that, obviously the multidisciplinary teams is hugely important. Big studies, multi-site studies are important. And I know that these are not the types of things that get funded or get published, and people are frowned upon the Me Too research, but we need it. We need that reproducibility. We've seen far too many even trials get too far down the road because it was based on one cohort that they didn't realize had inclusion criteria that only selected a niche group as opposed to something that's more globally applied. And that includes trying to have worldwide studies instead of just national studies. So we need more big data. We need more big collaboratives to tackle some of these issues. Super.
Brian, anything else to add?
Yeah, I echo that. It takes a village. It's not going to be solved by one technology or one omics. And the microbiome is relatively untapped. I mean, to think about all the different phyla and taxa and strains, and to understand all the different enzymes that they produce. So what metabolites, what bacteria produce what metabolites, and then when it's one thing if there's a single strain in culture, but when you're in your gut and you have all these different strains working in collaboration, it's a whole new story. So we're just scratching the surface. There's lots of work to be done. I think the field is doing great microbiome research, and we're learning a lot. Metabolon, as I'm sure other metabolomics companies are involved a lot in trying to, more fully annotate, microbiome-derived metabolites and how these things interrelate with each other. So I'm excited to see what the field brings in the years to come.
Super, thank you, Brian. Like I said, I thank you guys for sticking around for even though we overran the hour quite considerably. But it was an amazing experience to hear you all speak, and this work and field is so interesting. For those of you who are new to, this is the first webinar you've attended, the microbiome series is new to Frontline Genomics and was kind of put together as a result of the Festival of Genomics. We have some microbiome talks on there as well. I'll send a link for anyone who wants to listen to those in the meantime between now and next week. It's free to attend, and everything is on demand at the moment up until tomorrow evening. So if there's anyone who wants to just kind of catch up on those, you're more than welcome. But yeah, we have another webinar same time next week. Hopefully we'll keep to time, so please do come back. And again, thank you to the speakers for joining us, and have a lovely day, afternoon, wherever you are. Bye-bye.
Bye. Thanks, everybody.
Bye.